for joining us. It's my very great pleasure to introduce Lorin Crawford, who's a principal researcher at Microsoft Research New England, as well as an associate professor of biostatistics at Brown University. And he's going to be talking to us today about variable selection and prioritization in Bayesian machine learning methods. And without further ado, I will hand over to him. Thank you very much.
Thanks so much, Paul. Thanks so much for everyone for being here and attending. Again, my name is Lorin Crawford. I like to start off every talk by kind of going a little bit over like myself and kind of my research program, if that's okay to begin with. I think if you looked at like my lab's website, you might see that we kind of do a lot of different things. You might wonder like, what is it that this person actually focuses on? I like to think about statistics as kind of like this vehicle that I use to kind of explore different areas and domains. But I was co-advised with someone who went through my PhD in pharmacology, cancer biology. And so there's a huge biological undertone to a lot of things that we work on as a motivating factor for a lot of things. I can tell people I'm not a statistician that builds really fancy hammers and then goes out and look for nails. I actually really care about the specific biological problems that we are working on and trying to solve. And so at the very core center of this is statistics, and my group has grown recently to explore other areas, including problems in physics, neurosciences, as well as socio-technical type problems in society, which has been quite fun to work with. But at the center of this and the statistics core is this general theme of dissecting phenotypic variation. I'll get to that in a little bit. We build modern machine learning type tools in order to enable their theories and interpretations to be related back to genomic principles. So under all that kind of diagram, this is the kind of theme that kind of drives me as a researcher. Like I just mentioned, modeling complex variation across complex traits is something that is a huge reoccurring theme in a lot of the papers that we end up writing. And so, you can think of complex traits in many different ways. You can think of these as measurements across different bird beaks across different species, or you can think about this as heel bones across different primates. Today, we'll focus on a lot of human variation type traits and traits coming from mice, and a lot of the examples that I'll kind of show. But just to kind of get an idea of what I mean by dissecting phenotypic variation. So, any trait that we have, it could be height, body mass index, we could make that pie, I could take that variation, treat it as a pie, and we could break this down to different components. Okay, and so a lot of complex traits, there's a part of that phenotype that's broken down into part of it that's genetics, and part of it that's being driven by one's environment, right? Now I can take that genetic component and I can break that piece down into other further components. And so I can dissect that genotypic variation into two more parts. You can think about an additive genotypic effect, this notion of gene A's effect plus gene B's effect, or I think about this as a nonlinear effect. And we can think about nonlinear effects being in two different classes. One being what we refer to as epistasis a lot of times. So this notion of gene A's effect times gene B's effect, or gene by E environment, right? Like this notion of gene A's effect and how it's being interacting with one's given environment. Today we'll focus on the former rather than the latter, but this is the notion of how you can think about dissecting variation, and you'll see this theme pop up throughout the talk. So the data that I work with is we'll keep it really simple for today. We'll think about this notion of I have some trait of interest. Again, this could be crop yield in plants, height in humans, and we'll have some genotype matrix that's available to us, right? So we can think about this as some high dimensional matrix. N being the number of individuals. A lot of times in biobank studies, as you guys might be aware, we have hundreds of thousands of individuals, and then P being number of, in this case, SNPs. We work with a lot of SNP chip type data. So these data sets that are encoded as 0, 1, or 2, according to some like minor reference allele, right? And so, P could be on the order of millions, depending on how one has been able to impute their data. And so taking a little step back, to stats 101 maybe is, linear models are actually quite well used in genome-wide association studies and EQTL mapping-based studies, right? And so I like to show the slide for a few different reasons. One is to show the idea of this notion of a high dimensional modeling problem that we have, right, where P is many SNPs. P is also often much larger than the number of individuals that we sampled, but also this notion of in linear models is at the basis of these two fields, and what kind of drives their utility is this notion of like these beta J's, these effect sizes for all of our features in our data set, really lend some notion of evidence for association of a given locus, right? So what I do is I estimate these beta J's, and I can do a set of downstream tasks, right? After I have some notion of like how much an additive effect is for every single genotype in my data set, right? And these downstream tasks kind of fall into two basic categories. A lot of times, if you were to kind of take the papers in, at least in human genetics, and kind of split these split these papers into two different camps, right? The first is notion of phenotypic prediction. And this is actually a really tough problem. You could take this notion of I've observed a set of traits in genotypes in one population. I train a given model, and I try to understand whether or not that if I could take that model, then use that for prediction-based tasks for a set of other individuals, which I haven't actually observed that trait, right? So you think about this a lot of times with polygenic risk scores are a really popular thing right now, where I try to assess disease risk for certain individuals, right? And this is a really tough problem, actually, and it's a tough problem for a few different reasons. A lot of times what people have shown is if I train a model in individuals of one ancestry, and I go and test on a set of individuals of another self-identified ancestry, right? My prediction performance can be well off. And as that can happen for a few different reasons. One is this notion of, I'm only looking at these marginal effect sizes, right? In human genetics, where I might be missing part of the variation that's not really explained from these like marginal estimates. There's complex correlation structure, right, between the given SNPs in our data set. So features have these really complex correlation structures that actually differ across populations. It's quite interesting. And this other notion of, I might not actually observe the direct causal effect all the time. So there could be settings in phenotypes where I've measured in the generative model for a trait, there's a causal feature, but in the data set that I received, I don't necessarily have observed that exact causal SNP. I've observed a SNP that is only associated or correlated with it. So that's what we call this notion of tagging. So I don't actually have the causal features directly in my data set. That's another complication. And so phenotypic prediction in this space is an ongoing task that people are trying to fight. And this particular task will focus on or in this particular talk, we'll focus on a second task here, which is this notion of association mapping and fine mapping in human genetics. So this is this notion where I have these beta J's that I've estimated, and I'm really trying to understand some statistical significance for each feature in my data set, right? There's some level of association that is contributed to them. And this really boils down to this notion of testing a null hypothesis, right? I estimate these beta J's, their standard errors. I could do things like derive p-values for them, right? Then I can plot to understand enrichment across a given genomic region. So on the left-hand side here, what you're seeing is what we call a Manhattan plot. For those who may not have seen these before, each point here is a given SNP across our 22 chromosomes. And this is for an autoimmune disease. So you have this like nice spike in what we call the HLA region. A spike on the end of this plot means there's an enrichment at that given genomic location, right? And what's really cool is I can then go in and say, okay, there's an enrichment around this genomic region. I can go in on the right-hand side and zoom in and say, okay, which genes are then encoding in this particular region for downstream analysis. Okay. Now, everything I've kind of mentioned to you kind of centers around this notion of linear models. And so what's interesting about my group is my group tries to think about things past additivity, right? And so we really focus on this notion of black box statistical methods, right? This notion of, machine learning methods that have been really well suited actually for prediction-based tasks in many different application studies. The issue with them, as many of us are probably well aware, machine learning methods are also often tagged as being these things that are black box. And really black box just means that I really have no idea what's kind of happening in the context of these algorithms. And so one can think about this notion of, I put an input into an algorithm with this notion of I want to kind of study or predict a given output of interest. But the black box part kind of means I don't really understand. There's kind of haziness centered around like what's happening within the context of this algorithm. So, and it goes as output, but I don't really necessarily know how this black box is upweighting or downweighting or transforming these features in a way that's allowing me to really see these really nice predictive estimates, right? And so a big research goal of mine is to provide interpretable ways to summarize or explain or interpret, right, the importance of input variables in these kind of non-parametric technologies, right? In this talk, we're going to have our inputs be genotypes and our outputs in this talk. Now, going back to my notion of like my motivation of modeling phenotypic variation, like linear models are really nice because again, you have this nice interpretability, these well-founded notions of statistical hypothesis testing in these spaces. But the issue with them is they only see part of the pie, right? I only get to really model these additive effects, these environmental effects, and I kind of miss this portion of nonlinear variation, right? Whereas black box models kind of see the entire piece of the pie, but I don't necessarily have interpretability. So another way to kind of summarize my previous slide is to say, I want to work with black box models and get the benefits that I have on the right-hand side, and then figure out ways to do the same type of variable selection-based tasks that one might think about doing that I can do on the left, right? And so that's a huge methodological motivation. One way to say this for those in the room who are statisticians is smooth nonlinear functions are often more predictive than linear ones, right? But variable selection is much easier with linear regression. For those who may be more focused on areas of genetics, what I might say to transform that is, nonlinear models are often much better for tasks like genomic selection or phenotypic prediction, but linear models, as I have been motivating us thus far, are much well characterized for things like GWAS and QTL mapping-based studies. And so one big focus I've really been thinking about really since my PhD, and has been a huge focus on my lab group, is basically taking us on this road to interpretability, right? So taking these black box models on the left and figuring out how to add features to them so that we can move towards things that we consider in our everyday statistical life as being more interpretable, right? And we've been doing this now recently, not just with supervised based methods, but also unsupervised ones, right? Where you think about this notion of taking in the feature importance and then moving towards things where we are able to do things that are a little bit more well calibrated, like identify posterior inclusion probabilities, Bayes factors, and these type things. And so we'll focus on supervised learning methods for today, but I just want to kind of mention and kind of highlight, just for those who may be thinking about ways that we could all personally work together, is that, we were thinking about this in many different contexts. So what do I think about the notion of components of an interpretable model, right? Like what kind of allows me to start on the left-hand side of the black box methods and kind of move towards interpretability? Well, I kind of break this down to three different components, broadly speaking. The first is this notion of having an underlying motivating probabilistic model that's involved. Then there's this notion of deriving like an effect size, or this notion of a regression coefficient, right? Like, so the thing that makes, again, that linear models at the center of that slide I showed earlier is this notion of these beta J's are kind of at the crux of what we're able to do, particularly in human genetics, right? And then there's this notion of deriving a statistical metric that takes and summarizes marker significance, right? So not only am I able to derive some effect size, but I'm also able to estimate how important these given features are. Right. And so these three components will start to break down, and we'll break these down into two different ways that people normally think about them, particularly in machine learning probabilistic methodologies, right? So there's first notion is the notion of a post hoc approach to interpretability, right, or explainability, right? Variable importance or prioritization is another couple of terms that one might have heard, where you want to define, you fit a really complicated model, then I try to figure out what that model has learned, right? Post hoc, right? And so here we're going to define this notion of an effect size analog for nonlinear models. I'll talk about a significance measure that we created within my group, and then I'll go over simulations and real data applications in that space. The second part of the talk, I'm going to talk about this notion of neural network architectures that are governed by real world evidence or real world knowledge, right? And here we're going to focus in the context of biology, where we're going to leverage the types of hierarchical structures that happen within cells and molecular units to derive what we call biologically annotated neural networks, or bands in this case. And in this case, we'll talk about the scalable approximate Bayesian inference that we use to fit the bands models. Then I'll show some really cool simulations of real data applications in that space and kind of give you a few takeaways that I've had from working on these types of models. So let's dive right in. Let's start with this notion of a post hoc approach for interpretable ML. So very broadly speaking, let's think about let's relax our notion of having a linear relationship. So not necessarily this X beta, where I'm thinking just about the additivity across different SNPs and their coefficients to give rise to a trait. Let's now relax that assumption. Just imagine that I have some nonlinear method where instead of the x beta now I replace that with this f of x term. I could think about this f of x as being some function, some functional transformation of my data in maybe some nonlinear way. Here, what we're going to do is we're going to specify, we could put a Gaussian process type prior or specify a prior over this function space here. Or for those who may not have seen these before, a GP is normally defined by two key components. The first is like a mean function. Right, in the context of this talk, let's just imagine that that is like 0. And the second is kernel covariance function, right? So we'll allow all of our nonlinearities in this model to be derived from that nonlinear covariance. You could think about this as like a similarity measure for those who may not know. So a really classic example of this case is to use a Gaussian kernel function, right? According to that equation at the bottom. What's really nice about the Gaussian kernel is you can actually take the Taylor series expansion of the Gaussian kernel, kind of show that you're enumerating all these possible pairwise and nonlinear interactions implicitly within the context of that kernel. And so again, you can think about this as like, I can derive, once I choose my kernel, I can have this kernel matrix or covariance matrix K, which is basically assessing the similarity between any two samples genotypes, right? So kx1, x2 is in this nonlinear way is determining some notion of similarity between those individuals' SNP data. Now, the nice thing about the kernel function is it allows me to now specify these nonlinearities or make really nice predictions. But with this notion of what we call the kernel trick in the literature, there's a little bit of an issue, especially if your goal is for interpretability, right? Or variable importance. So, I had this original high dimensional P space in our case where I can do these linear models, but what I actually want to do is I can specify this kernel function. What that does is it allows me to move to this n-dimensional nonlinear function space for which I can do really nice prediction based tasks. But if my goal in biology is to understand within the context of this nonlinear model how SNPs are being upweighted or downweighted, there's no real notion of, translating this model's fit back to my original p-dimensional space, right? And so from the context of our classic notion or idea of variable selection, that idea is kind of lost when we do this kernel trick transformation, right? And so we're going to stick with these Gaussian processes for a little bit, and we're going to treat those as our motivating probabilistic model, but we're still missing the notion of an effect size in these methods, okay? So that drove us to think about this notion of the effect size analog. And so to give everybody an idea of how the effect size analog works, actually quite simple. And I'm going to do this mirroring what we do in linear models, right? So in linear models, what one can do is I can think about an effect size, right, as this notion of a linear projection of my data Y onto the column space of my covariance, right? So I project Y onto the column space of X, and that gives me these like nice little beta hats, right? And I can think about that projection in a lot of different contexts, but for the sake of this talk, let's think about this in the least squares type of way, where I think about X just taking some generalized inverse of X and projecting Y onto via that linear transformation. Now, nonlinear models, I can do something very similar, where instead of projecting Y into the column space of X, I project F, my smooth nonlinear function, instead onto the column space of X, right? And we can think about these beta tildes now after I've kind of trained and learned this, fit this model with this now smoother function. And these beta tildes as a effect size, like I said, an effect size analog to what I might have seen, might have done with linear models. I can choose any projection that I might want here. Here we'll think about this using just a standard, for the sake of this talk, another standard like least squares linear type projection. But I do want to mention that there are other groups that have been doing kind of cool work with this, where you can think about different projections between some smooth function and your X to kind of pull out different features that one might want from that given function, which has been quite cool. I'm happy to send people's work on that if people are interested. So, I went to Duke for my PhD. For those who may not know, Duke University is cultly Bayesian. So let's think in a Bayesian type of context here. Let's imagine I take a weight space view on Gaussian processes here. We'll specify some priors for the rest of our models. And as the key thing about this slide is, as soon as I draw new estimates for my function F, as long as I've set up some projection that's deterministic, I can deterministically figure out or draw implicitly some draw for beta tildes, right? And if I do this many times, I can start to do really cool things where I can specify, posterior predicted distributions and all kind of things for these beta, which are actually really cool. So I just want to show everyone really quickly that this actually works in real data, because one might be saying, like, what can I actually do with these? Well, I can use these beta tildes for any kind of task. Let's think about this in the notion of genomics selection really quick. So phenotype prediction. I like this data set a lot, actually, because this data set comes from the Wellcome Center for Human Genetics. I have 2,000 related mice in this data set, 10,000 SNPs. This data set is really nice because it's broken down and it has 129 different traits that you can break down into 6 different broad groups, all with different levels of additive types of variation and non-additive types of variation. The key thing I want you guys all to take away from this slide is that, if my effect size analogs are working correctly, then they should produce the same sorts of predictions that a support vector machine or some nonlinear model will make, right? Because I fit this thing with a nonlinear model and derive them from the smooth nonlinear function. And they should be more stable, especially with phenotypes driven by non-additivity than linear models. So things like lasso, ridge, linear LMMs, we should do better in these methods, right? What we're going to do is we're going to do these 80-20 N analysis interfamily splits between these mice, and I'm going to do this 100 different times, and I'm going to plot these mean squared errors of predictions across these different methods, right? So we fit our GP, we derive effect size analogs, I use those effect size analogs for predictions. Here are the overall results. So again, MSBE lower means better here, and you kind of see the overall summary that I was hoping to see, right? Where out of 100 different times, the SVM and GP are always kind of the best given models. That's what that optimal line means at the bottom there. And you can see the SVM and GP, they're pretty similarly closely in terms of performance clustered together, and then you have the linear models clustered with themselves. This plot is helpful, and I like to show this, but a better plot to kind of show this how performance kind of varies across all of these traits. So what I could do is take this table and just give you a broader view where MSPE, or mean squared error prediction rate, is on the y-axis. Our 6 broad groups are kind of labeled there across all 129 different traits. You can see the GP and the SVM, which are these red and purple lines, kind of track really well together across on the bottom there. Where for certain traits, there are spikes where the linear-based models kind of drop heavily off in performance. What's really cool about these data sets is you can actually attest to whether you can actually see and assess whether nonlinearity is actually driving the variation for these traits. So what I do is across all 129 different phenotypes here, I run a variance component method where we break down the types of variation that one might see, whether that's additive, pairwise interactions, third order interactions, or things that are driven by the common environment across these mice. And what's really nice is we could plot these. And across these different traits, actually, very rarely actually is additivity kind of driving the variation of these traits. So at the top, you see additive effects, you have pairwise, third order, and common environmental effects. And the spike there means how much variation is being driven by that particular type of effect. And you can see for some of these traits, common environments, third order, and pairwise interactions are actually huge drivers of these traits. And these actually map really quite nicely on the previous plot, where a large dip in performance on these linear models are for phenotypes where these other components actually matter quite a bit. So these effect size analogs are really cool. And what's nice about them is they give us these notions of effect sizes for our regression models, right? Now, the issue with them, however, is that you can't use them for variable selection. They're just weights, right? So they're the same kind of predictive weights that one might think about using them for. So they don't really give you a notion of ranking here. And so that kind of begs the question, well, what do I do? I don't necessarily get a natural p-value or Bayes factor inclusion probability that one might see in other areas of stats. So what do I actually do if I want to think about variable importance in this way? And so we came up with a metric to kind of do this. And this is derived from this notion of kind of information theoretic ways of thinking about how we might rank players on a team. And so I'm going to show this in the context of sports. This is going to be in the notion of basketball. I maybe should have picked football or something for this particular audience, but let's think about this in the context of ranking players for this particular team. So we'll take the 90s, a Bulls team from the 90s for a basketball team. And the concept of this, I hope, is very intuitive in the sense of when I have an entire team, I can think about each player on this team as being part of a network, right? And so together, that network has some sort of information when it's all of its components are together, right? And the effect of all their components are together. Now, if I want to assess how important any given node is in the context of this network, what I could do is I could figure out ways to nullify its effect, right? So in other words, let's say I take number 30 and I say, I'm going to put the way I can figure out how important number 30 is, I could put number 30 on injury reserve, right? And then I could say, how intact is the information in my network with number 30 around versus when number 30 is not around, right? In other words, I can kind of measure the distance between, if you think about this, like this notion of performance, what happens when number 30 is on injury reserve versus when number 30 is on the team, right? And if it's not a huge shift, then I might say that, well, number 30 is not really a key player in explaining what's happening on this team. Now, let's say I take another individual. For those who may not know, this is Michael Jordan. I might say Michael Jordan is not playing basketball anymore because he decided in the 90s he was going to play baseball for a little bit. Now, if I lose potentially the best player of all time, I might see a bigger shift in information, right, than when number 30 wasn't around, right? And I could take this metric and do that for every single player on my team and rank every player's relative importance relative to the rest of the team by looking at that difference, right? And I can do this by basically using KL divergences, right? We can specify this in this way where I can summarize the influence of any given feature in my data set on the rest of the features by measuring the KL divergence between the conditional distribution when that player's effect has been set to 0 versus the marginal distribution when I've marginalized over that player's information, right? So the KL divergence being equal to 0 here can be interpreted as like that key variable is actually not explanatory relative to the others, right? There's notion of that if that distribution doesn't really shift at all, I haven't really lost any information by not having that player. Or alternatively, you could say that the KL divergence is 0 if and only if those two distributions, right? Now, in this particular paper at the bottom here, we do this thing where we scale by the sum of these to kind of derive what we call a relative centrality metric. What's really interesting about rate is rate has a really interesting null model where under the null, rate's assumption is that all players are contributing equally to this network, right? This notion of if every player shifts the distribution exactly the same, then no player is a key player relative than the other ones, right? So this notion of like this 1 over the number of features I have in my data set really signify the difference this notion of I don't have any stars, right? And the alternative is that I do have stars. Some players are much more key to explain the variation in that network relative to the other. So really quickly, let me kind of go over a quick simulation study. We'll focus again on a thing where I have 2,000 samples and 25 genetic markers. I'm going to choose 3 of those markers to be causal just for the sake of demonstration here. For those who may be aware of the Chicago Bulls, these will be my Jordan, Pippen, and Rodman, my 3 stars of my team, and the rest of my players will be considered bench players, right, with no effect. What we're going to do here is I'm going to simulate these data. I'm going to fit a GP, estimate an effect size analog, and then I'm going to run this rate metric, and I'm going to rank all of my players according to that rate metric and then show you the difference. Okay. And so here are those results. On the bottom, you have all of my 25 covariates. My rate metric is on the y-axis. And then what you're seeing is I'm identifying this, my 3 stars. That red line at the bottom represents that 1 over p line I was mentioning, this notion of like I have no key relative players anymore, just only bench players. So you can see I identify my Jordan, Pippen, and Rodman covariates actually really well reliably, right? Now, what I can do in this model to kind of give you a notion of what I mean by relative centrality is I could say, well, what happens if I take any of these individual players and I put them on injury? I put Jordan, let's say I take Jordan out of the game, and then I also condition everyone else's effect being equal to 0 with Jordan's, right? So Jordan's always out, and I'm gonna just rank all of my players after that. What I can do is I can say, well, if I always hold one variable's effect constant and then run rate again, a really interesting thing happens where I still identify the 2 features that are causal, but the relative importance of all the other features has gone up, right? That's like saying I'm missing my star player. My star player is no longer on the pitch or on the court. All my other players those points in that performance have to be made up somewhere, right? Now, the importance of every the other players are going to go up, although the variables go up equally, but that's because they're all noise variables, right? No one is more important than the other, so that burden is kind of distributed evenly across all of them, right? I can do the same thing again, where I now hold my second key variable out with my first and then rerun rate again. You can see these variables kind of get tighter and tighter around that 1/p line. And I could do this again until I have like almost no key variable. Now, again, on this proof of concept, I might say, as a sanity check, okay, what happens if I took a random variable out? What happens to the distribution of these rate values? Where you can see my conclusions are kind of the same. And we can also say, well, what happens if we simulate data with just noise? Well, I can do that, and you can kind of see everyone kind of hovers on these two different draws. Everything kind of hovers around that 1/p. Now, rate's not perfect, and let me kind of get to this in a second, but let's really quickly revisit our mouse data set, where, again, I have my 2,000 mice. Out of the 129 different traits now, I'm going to run the GP, the effect size analog, and rate, but only on one trait. Let's just look at an HDL for this context. And I'm going to compare what happens between an additive univariate single SNP test that we typically do in human genetics, and I'm going to compare that to what one might get with a GP in this rate type model, right? And I like this trait for two different reasons. So on the top, you have the performance from the Gaussian process. On the bottom, you have the context from the results from the single SNP model. And I like this trait for a few different reasons, because there are known additive effects that are actually quite strong on chromosome 1. And you can see actually really nicely how the single SNP additive model and the GP really nicely overlay and identify in that given region, identified by that star there. And there are also regions where non-additive effects actually have been functionally validated for this given trait across different studies. And those typically happen on chromosomes 11 and 12 are huge regions there. You can see how nicely these are identified with the context of this GP, but are actually quite missed for this single SNP model, which is actually kind of interesting. Now, this plot actually identifies the key issue with rate, right? So rate is what I call a variable importance metric. It allows you to rank your variables, but it doesn't allow you to do things in statistics, like if you care about like creating credible sets or identifying some significant cutoff for where I might think about things. That 1/p line is not that informative for high dimensional data sets, right? As p goes to infinity, and the key issue with rate is that 1/p line basically goes to 0. And if I think about that as an acceptance threshold, now I'm accepting all variables as being important, right? And so rate just helps you rank variables, which you need to do something downstream in order to actually identify, some thresholding values for which you choose to create these credible or causal type sets for validation, right? And so those are key issues with rate. I'm sure people are going to ask me the questions. I'm happy to talk about all the limitations of this method in a little bit, but I want to kind of use it as a motivating factor where, the key thing that I think motivated us for the second half of this talk are some of these actually limitations, right? And so I want to move to the second part of my talk as time continues to escape me, where, rate gives me this notion of these three different components, right? But we have these like natural limitations of, I would like to, on that spectrum I kind of showed earlier, I would like to move a little bit closer, not just to have a model that's explainable, but have a model that's more interpretable and that looks much more similar to the types of outputs that one might get if I were to think about running like a linear regression type. And so that brought us to this notion of using neural network architectures that are kind of governed by biology, right? Now, for those who may not have seen a neural network before, I could use a rate model in a neural network, right? And I like to call machine learning just fancy linear algebra. They're just fancy hierarchical nonlinear models. So we could write a neural network in a very similar fashion to like the GP type that you saw before, or this is what we call a fully connected neural network. Every preceding neuron in the preceding layer is connected via some weight, some theta, or some W in the next layer, right? And so everything here is fully connected to each other between different layers, right? I can write my function that my neural network learns, right, as a linear combination of these nonlinear hidden activations, these H's here, and there are these W's. Now, if I made this a probabilistic or Bayesian model, I can get a posterior distribution over F, and then I can learn the effect size analogs for them, and I can also fit rate. What I want to kind of do for this talk is or this part of the talk is move away from that and think about ways to actually get closer towards more like a posterior inclusion probability type models that I can start to think about sparse priors and think about variable selection in a more classical way, right? And actually, what's really cool about biology is you can start to do that by leveraging this notion that there's a hierarchical nature often to enrichment studies. So again, this is a Manhattan plot. Each point here is a dot. Each point here is a snip. And what's really cool here is I can annotate along the genome certain genomic units that I know are encoded in that given region, right? So I know which genes are encoded across my given genome, right? So it's kind of like this hierarchical nature where SNPs kind of feed into genes, right? I kind of have this notion of that along the genome. What that actually means is that in all of my biological studies, I get these nice predefined groupings, right? Where I know I have a gene, I understand what chromosome that gene is on, and I understand the start and end position windows along the genome that the chromosomes are. So I know which SNPs fit within that group as an annotation. Well, I can use that information to inform network architecture, right? So stay with me really quickly. At the bottom here, another chromosome, or the Manhattan plot. Again, each dot is a SNP, and each of these red points is a given gene. If you think about it, these SNPs are being fed into genes, right? Well, if I turn this Manhattan plot on its axis, right, SNPs being fed into genes along the genome, this sort of looks like a partially connected neural network, right? Where on my first layer, I have SNPs, and SNPs are only going to be connected to a hidden layer if they've been annotated for the same genomic unit, right? So in this context, this is partially connected. Again, weights only are non-zero in this case if they've been annotated for the same particular thing. And I can do this along the genome. What this does now is it makes my hidden layer just not a hidden layer. It makes this now that every neuron in this case is a gene or can be interpreted as some SNP set, some SNP grouping that I use my annotations for. And now each of these weights aren't just weights, they're not just arbitrary thetas anymore, right? They're an effect size of how that given SNP contributes to the context of that given gene, right? Or how that gene now for these W's now contributes to the output of my phenotype, right? So now I have interpretability, or I have some interpretation that I've imposed on the model because of those annotations throughout the specification of my network. So this is what we call bands, and bands is actually quite easy to do from a general perspective. You know, I have some groupings, again, my SNP sets, the chromosomes, start and end positions, the SNPs that fit within those chromosomes or within those chromosomal windows. And I could do is I could take panel A and I just translate that into what I would consider to be my partially connected neural network, where again, connections only happen if those annotations have been specified in that previous step, right? What's really nice about bands is now because every single weight has a meaning, I can start to specify specific priors that I want to attach to it, further infuse more biologically motivating fits for my model. So let's say I have some architecture of a trait where I assume that this trait's architecture is quite sparse, where I might have SNPs with very large effects or SNPs with no effect at all. I could translate that into like a spike and slab type prior. Let's say I have other traits where I might assume that the architecture or the distribution of SNP effects is actually quite complicated. So I might have a setting where I have large effect SNPs, no effect SNPs, and then SNPs that have like medium or intermediate effects. That's going to be specified as like a more of like a mixture, right? And I can now specify this model in a hierarchical type of fashion, right? So each layer, so at the top, I have like obviously my full model specification of how my my phenotype is being modeled, but that's just again a linear combination of the hidden neurons and their weights and some bias terms, and the hidden neurons, right, are just a linear combination of the SNPs and their weights and some bias terms, right? And again, I can specify different types of prior distributions depending on how I think maybe SNP level effects are distributed versus SNP set effects, like gene effects are distributed, right? Bands is quite flexible. So on the previous slide here, I'm working on this with what we call individual level data, this notion that I've seen everyone's genotypes and phenotypes, right? For those who may not be aware, in genetics, we actually work with a lot of summary statistics sometimes because of privacy concerns, data distribution concerns, right? Where I work from more of a meta-analytic perspective. So that means that someone has taken your genes, your genotypes and your phenotypes. They've run their own regression model, and they'll release to the public like the effect sizes and the p-values and stuff like that. Bands also can work from that standpoint, so we can work with summary stats instead of just having individual level data. And one thing I've been really interested in as of late, what's been really cool, is Bands also works with multiple phenotypes. So let's say that I not only want to do association mapping just from single traits, but I want to leverage the correlation across traits in order to have more power in identifying causal SNPs or SNP sets. I can do that by leveraging this notion of the correlation across traits in order to do so, and more like a matrix normal type of set. So what's the key thing about Bands is Bands does two things. One, it allows us to do simultaneous multi-scale genetic inference. There's this notion of posterior means of the weights for non-associated sets will be pushed towards 0 in this model. And again, we derive now more classical summaries in like the rate type model, where I now get things like posterior inclusion probabilities, again, at multiple scales. So I'm able to identify SNPs that are important, right, according to the posterior inclusion probabilities for these thetas, as well as on the gene level for those subsequent layers, right? And allows me to do multi-scale inference that way. One might think, why are multi-scale inference models like actually really useful and quite interesting? Bands is an interpretable model because it gives me all these three components. But why Bands is cool from a multi-scale perspective is because you can imagine that different types of phenotypes have different types of models that might fit my idea of what is replicable across different populations of people. So there might be a SNP model where I might say the same mutation is a driver across all traits for all people, right? So that means that all of us for a given trait have the same mutation that's driving our phenotype. There might be some traits where they fall more under the gene model. So the gene model means is that we all have different mutations driving a given trait, but those mutations all affect the same gene. So we might have different features that are causal across all of us, but the same gene model is actually the thing that's replicable across all of us. That's the key thing that's being dysregulated, right? And then there's a pathway or network model, which says that we all have the different mutations and we all have different genes that are driving our phenotypic variation, but those still may might dysregulate or regulate the same mechanism, right? So our metabolism might be off, even though the different genes that are being dysregulated are the things that are not the same. And Bands gives you a way to kind of look at these different pathways or these different types of scaled models all at once. Right? So really quickly, let me go over a simulation study. The key thing that I kind of wanted to know is here we take chromosome 1 from individuals and self-identify European ancestry. This data set has, we're going to limit ourselves just to chromosome 1. So we have about 37,000 SNPs, about 350,000 individuals. We map all SNPs to these different genes according to this database. So here we have 1,900 or so genes. And we're going to assume a heritability of like 0.6. The key thing about Bands is Bands should be able to identify both causal SNPs better than both causal SNPs as well as SNP level methods and SNP sets. So it should be able to do both those things at the same time. So we're going to compare Bands to models on both these scales. We're going to do so across two different types of architectures. The first is going to be a sparse architecture. So again, I have my two different traits. Sparse architecture just means I have a few SNPs driving the variation of my phenotype. That means that every single SNP is going to have a large effect size. Whereas a polygenic one, it means I have many SNPs, right? So you can think about that as having smaller effect sizes. So what you're going to see in our results on this next slide is that the sparse architecture is easier because the effect sizes for every single causal SNP is larger, right? And that's kind of what you're seeing here, where here's the performance of Bands and some SNP level type models. But overall, the general theme is that polygenic architecture is harder. But you can see how nicely Bands kind of performs with respect to competitors on this space. We could do the same thing for the identifying core genes. So does Bands actually rank genes better than other methods? You can see this is actually how well it does across its competitors here. The key thing that we've been working on recently is trying to improve the runtime of Bands. For those who may have noticed on the previous slide, I said that Bands is fit with the variational EM algorithm. This ER algorithm is fine because it scales relatively well. It's not perfect. One, it doesn't allow you, because of certain approximational properties, it doesn't allow you to derive credible sets from these posterior inclusion probabilities. The other thing is that there can be downward bias in the sampling of the effect sizes or those weights for our given model. So what you can do is say, can I actually recover the true model from the generative model I used in my simulations? When the architecture of traits is quite simple, so meaning sparse and additive, Bands is able to do that with the variational EM. But like in many variational EM algorithms that people have seen and throughout ML models, there's a downward bias where there's the undersampling of these weights and these approximations as the architecture gets a little bit more complicated, right? And so these are so the EM algorithm is not perfect. Again, happy to talk about the limitations of Bands and the question. Really quickly, I just want to show what happens in real data. So we look at the Framingham Heart Study here in New England, and I can also compare this model to, the results to the UK Biobank for 10,000 randomly sampled individuals. We're going to look at HDL here. I just want to give a quick view of what kind of happens. Again, at the top, we have the Framingham Heart Study, where each plot point here is a SNP, and then you can see in the top, these are the genes that correspond to being enriched for these given genomic positions. A star here means that these are replicated across these two data sets. And so we do this, we do a lot of different analyses in that paper that's at the bottom there, where I think there's like 60 pages of supplementals if you're interested in going through this at all. And one last thing that I think I want to highlight is what's really cool about these gene models is you get very nice architectural differences across different traits. So here we take triglyceride levels. So I take the enrichment that I see from the Bands model, I take those genes and I say, how do these kind of differ across networks, across different people? Genes give you a better notion of resolution of what's shared and not shared across different people. And so here's a network of enriched genes found in these 3 different populations, people with self-identified European, East Asian, and Oceanian ancestry. And you can see is how nicely there are parts of these, for this particular trait, for genes that are commonly found across things, but also things that are quite unique to certain populations. And so genes give you this nice hierarchical resolution, again, further enforcing the utility of a Bands-like model. There's many stuff that we're doing in the group, and I'll just kind of wrap up here. We've been extending interpretability methods in many different ways. This includes developing alternative effect size analogs that you saw before, as well as extending Bands to multi-omic data set sources. I've been really interested in doing machine learning for small n type problems, and that's been quite fun. On the right-hand side here, what you're seeing is what we call the single effect neural network. It's a neural network that allows you now to actually develop these credible sets. For those who may be aware of the genetics of the community, there's this model called SUSY, which allows you to build these credible sets. And we kind of extended this method to think about things in the context of neural networks with nonlinearities. I've also been interested in running these like rate type models for studying shape variation for traits. So you can imagine a context where I want to identify physical locations on like a brain tumor, for instance, that are hallmarks of given mutation statuses. So we've been doing these things where we do like variable selection on 3D surfaces, which has been kind of cool. All of this understanding of trying to understand when shape variation is correlated with genotypic or phenotypic variation, right? So that's been actually quite fun. Just to give a little plug, the single effect neural network, this is a paper that's out in iScience. What's really nice is you could take some of the things that we learned from the Band framework and think about other ways to specify and derive more posterior inclusion type models, probabilities for neural networks. And very lastly, we've developed these cool variable selection 3D frameworks where you can input 3D shapes from objects, and these are teeth from primates. You can encode summary statistics that basically encode their geometry and topology. And then what you could do is once I have these nice curves, I can run a GP and rate model to identify the pieces of the curves that are most variable between the two groups. Because our transformation from shape to summary stat is invertible, once I identify the significant pieces of those curves via like the GP and rate, I can project those back onto shapes to identify the variation between the two groups. And so you could do this for anything in terms of like anthropology, like we did here, or clinical imaging and things like that. So we've been working a lot in this space as well. It's been fun. I'm happy to talk about that offline. So yeah, this is my talk and this notion of my road to interpretability. I definitely want to end by saying, along this spectrum, you don't have to always fight to get into interpretable models. There are a lot of contexts where explainability is best for certain contexts. So this is definitely a context specific journey along the spectrum. With that, this is an acknowledgement of many different collaborations and people. Thankful to the organizations that have supported us and given us money, to say that bluntly. And I'll close with some relevant references and happy to take questions.
Thank you very much indeed. That was fantastic. And yeah, if anyone has any questions, could you either raise your virtual hand or write something in the chat and I will read it out. Of course, better if you're willing to raise your hand and actually say your question yourself. But while we're waiting for people to write things in the chat or to raise their virtual hands, I will ask a question. So actually, I'm going to jump to David, see if I'm in turns to
okay.
Go on, you
Sure.
put your hand up, David, go ahead.
Hi there, thank you. That was a really cool talk. I just got, my CPU is dying, so I hope you can hear me.
Yeah.
I've got a sort of very niche sort of genetics question for these models. So for the
Sure.
the band models, how are you encoding sort of cis and trans regulatory elements? Is a cis element going in the ORF it's connected with, and then trans elements are just floating, or?
Yeah, it's a great question. So in this first paper, we didn't do any of that. So what you're going to see in this first paper is we just thought about SNPs being fed into genes, and then for the SNPs that were in like what we call like gene deserts, these genes that like hadn't been really annotated for anything, we treated those as their own specific node groupings. There are much more cool things you could do, like you just kind of mentioned, where you have more intricate kind of functional relationships and you kind of try to encode throughout the genome. We only thought about this individual 2-layer thing to begin with. There's one caveat I want to mention, which is as you get more hierarchical in your groupings, you want to really be careful about the notion of the groupings overlapping too much. So one thing that the variational EM will have a problem with is if their group overlaps way too much, the spike and slab in the variational EM is going to act more like a lasso than like a true spike and slab prior, where if I have truly overlapping or 2 overlapping groups, it's going to pick one of those 2 groups. And even if both of those things are true positives, it'll set one of those things to be 0 and it'll just select one of them. So as you get more intricate in thinking about how to group these things along the genome, that is one caveat to really think about that we didn't explore in this paper, but I have seen in further explorations of the model that I want to point out to people.
Thank you very much.
Yeah.
Gilbert, I see you have your hand up as well. I can't tell if you're talking, but I think you're muted still. Apologies.
Here we go. Sorry about that. That was a very fascinating talk. Clearly, your ideas and interpretability are very relevant. I was wondering, how will we know when the band model is inferior to other models? How do you propose to discover when the idea that you have on the hierarchical structures are wrong?
Yeah. So there are a lot of things to unpack here. So bands is not good in a few places. Let's think from like a data perspective side first. When you have low sample sizes, that variational EM is not that great, and actually those approximations that the model make will not perform that well. So that's one context. The other context is for lowly heritable traits that are highly polygenic. There's not a ton of separation between bands. Actually, in some of the examples in the paper, bands doesn't do that well in those models either. The other thing that bands are really susceptible to is susceptible to user definition. So going back to the data, is susceptible to user definitions right now, right? So that means that like bands only models whatever users specify as being these partial annotations. If those partial annotations are not correct, bands can actually identify a gene that you didn't actually, that is confounded by this notion that you didn't specify your boundaries correctly. Actually, we have an example of this in the paper. There's a trait, and I'm totally blanking on which one, where if I specify the gene boundaries according to the RefSeq database, I'll identify one gene as being important. But if I extend those boundaries to include regulatory units for that gene, I'll actually, for all genes, I'll identify another gene as being important. And that's the gene that people have identified as being like, this is a classic conundrum where people have shown that if I include regulatory units for this particular trait, actually, you'll misunderstand which gene you identified as being causal. And we actually replicate that finding in this paper. So that's another thing as well, is bands only takes in user data availability. I would love to figure out ways for, so another limitation where bands is not useful, I think I can go on about this, is if you want to actually use the bands model to discover new types of gene sets, bands can't do that. Again, bands treats these data sets as these annotations as fixed. I would love to figure out ways to maybe make these more of like a prior for which you can kind of learn posterior things over these annotations. We just haven't thought about ways to necessarily do that yet. But yeah, there are many ways where bands is not superior at all. Yeah.
Yeah, that seems sensible, so it does, and it's really roughly what I thought. Although I have to say I don't know anything about anything in detail about genetic modeling. Really, thank you very much.
Thank you.
Thank you. And Life, I see you have a question as well.
Yes. Yeah. Great talk. I was interested in this. How you get the effect sizes out of a Gaussian process regression model through a projection step. And I think you showed a plot there as well, where you said that you managed to get the effect sizes of interaction effects as well. Did you have that in your table? I think you did. And so my question becomes, you need to create this basis that you're sort of projecting down onto, right? So you have your GP and you project down to a basis. But the nice thing about the GP, right, is you never have to explicitly construct that basis for higher order interactions. And I was wondering how you did that, because that's something we are also interested in.
Right. So I didn't necessarily do this in my paper. So in the 2018 paper that's listed here, we construct.
Great.
We show that there's a you could use random Fourier transforms to construct like an empirical estimate of those bases, and that facilitates your projections backwards. The key limitation of literally everything that I showed today is that you get an overall association metric, but you don't necessarily know what's driving that association. So in the effect size analog and what we showed, you get an effect size, but you don't necessarily know where the effect size is being driven by additive effects, non-linearities, and in those non-linearities, what's actually pairwise, what's third order, There have been papers, and I can tend to see the last authors, the first author's last name I think is Wood. It's a JGCS paper where they think about how to construct specific bases where if I wanted to do this projection and pull out just interactions, this is how I might think about doing that. The limitation in our models is that we don't think about how to do those teasing apart, but that's something I would love to kind of explore if anyone's kind of interested in those kind of questions. But yeah, that's one thing that we are not able to do here.
Yeah, okay, yeah, because that is very interesting, right? But it's very hard because then you have to construct this gigantic feature space of all the pairwise interactions, for example, which is just impossible to do if you're in any high dimensions at all, right? Yeah, thank you so much.
Right, exactly.
I have a possibly naive question. So in the old days when people were interested in sort of variable importance scoring in GP regression, what they would do is this sort of automatic relevance determination stuff where you look at the inverse of the length scale associated with each of the features. Could that be used here, or is there a reason why that's a bad idea?
Oh no. So it can be used here. In the paper, we kind of talk about the differences between these two approaches. I think for us, what we were thinking about was high dimensionality of these data sets and thinking about like collinearities and how sometimes those types of methods are susceptible to these issues that one might see. The other thing that we were thinking about here as well is so some of those models or some of those approaches, particularly in the Bayesian context, struggle during MCMC with actually like fitting in estimation procedures, where it's like really hard to actually find a posterior range where you're getting reliable estimates and not have so much jump happening in MCMC. So we thought about all those type of limitations. And one of the in this model, I think, or in this paper, we compare the differences that one might imagine, because you could think about ways that I can say I'll fit like I can have like an inclusion probability of whether or not this length scale for a particular feature is set to 0 or not. We just thought about the scalability of our problem and fitting that to like a high dimensional data setting versus some of the data settings I think that those older models were fit to as part of the motivation for moving past that.
Fantastic. And I see Brian's hand up, and maybe we'll make this the last question in the interest of time, but go ahead, Brian.
I think it's a kind of follow-up question to leave. And when you fit these kind of Bayesian approximate kernel regression models with variable selection, you have an issue of either penalizing the kernel space or penalizing the input space. How do you reconcile that issue? Because sometimes you might be reducing the dimensionality of the kernel space rather than the input space.
Yeah, so in the kernel regression model, we do the dimensionality, right? We fit quote unquote like shrinkage priors in the kernel space. I haven't reconciled this issue yet. This is a really good point. We haven't thought about this notion of what sparsity in one space means and how that translates to sparsity in another space, right? Ideally, what you would love to do is understand like how, if I were to fit like a spike inside prior in kernel regression space, how those posterior inclusion probabilities translate to input space. We haven't thought about that. I haven't figured out a way to think about like that transferability. So I haven't reconciled that to be short with your answer. In this paper though, we do shrinkage and dimensionality reduction in kernel space with the notion that this might help in our rankings of variables in the original feature space. But theoretically, I haven't thought about those two at all. Yeah.
Can I just ask a final question, Paul?
Yeah.
How do you assess whether Phil Jackson is influential in the Chicago Bulls team?
How do I think about who's influential in the Chicago Bulls team? Yeah.
Phil Jackson, the coach. Because
oh yeah. He's probably super important, right? Like I don't know. I'm a Lakers fan, and I know he was infinitely influential for those teams, because when he left, we didn't win anything. So.
And on that note, thank you again. That was fantastic, and some really good questions at the end. Such an enjoyable talk. Thank you for joining us. And if you were here in person, we would be applauding loudly right now. So I hope we can. Some of us are sort of clapping virtually, but yeah, this is very much appreciated, and we really enjoyed your talk. And you may well hear from some of us subsequently who have sort of related interests, I think, if that's okay.
No, that's perfect. That'd be great. That's what this is for.
Fantastic. So on that note, I'll say goodbye to everyone. Thanks for joining us, and see you again at the next BSU seminar.